Hi, everyone. In this section, we're going to learn about the microscope, which is a very important part of microbiology. Microscopy lets us look at microbes that are too small to be seen with our eye alone. And this is so important for identifying pathogens. Again, pathogens are microbes that make someone sick. So at the end of this section, I am hoping that you'll be able to understand the units of measurement of microbes, the tiny, tiny units of measurement. You'll understand the parts and functions of microscope parts. You'll be able to describe magnification, and you'll be able to differentiate types of microscopes like light microscopes and electron microscopes. Okay, so just to remind everyone, microbes are very small. They cannot be seen with our eyes alone. So this slide is a bit heavy, so I just wanted to break it down. Just briefly, when we think of our eyes, I don't want you to memorize these measurements, but if I ask anyone, can you see a human with your eye? Can you see an apple? Can you see a tiny ant? Everyone would say yes. Can you see the end of a hair tip? Yes, you can. So a human eye can see all of this. Think of the smallest thing you can see, like a speck of dust. All of that our eye can see. Now let's take a speck of dust as an example. Imagine if you were to take a speck of dust or the end of a hair tip and split it 1,000 times, you wouldn't be able to see it with your eye alone. That's where a light microscope is important. So we call the units that a light microscope measures micrometers, and even smaller than micrometers are nanometers. And for that, we need a very fancy microscope that we call an electron microscope. Okay, so briefly about measurements. So bacteria are measured in micrometers, and here's what I mean by that. So the end of a pencil is a millimeter. That's like the end of a hair tip. We can all see that with our eyes. If you take a millimeter and you split that 1,000 times, that's called a micrometer, and sometimes it's abbreviated That's a micrometer, and that is a unit of measurement that we use to measure bacteria. So bacteria are in the micrometer range. For example, you don't have to have this memorized. E. coli bacteria is around 2 micrometers. And again, a micrometer is 1,000 times smaller than a millimeter. Now, viruses are even smaller than bacteria. Viruses are very small. If you take a micrometer and you split it 1,000 times, that's a nanometer, and viruses are measured in nanometers. For example, the HIV virus, which can cause AIDS, which means you don't have an immune system over time, is around 120 nanometers. So what I would like you to take from this slide is bacteria are measured in micrometers. Micrometers are 1,000 times smaller than millimeters. In order to see micrometers or see bacteria, we need a light microscope. Light microscopes are the traditional microscopes you see in lab. Viruses are much smaller than bacteria. They're measured in nanometers. And in order to see nanometers or viruses, you need an electron microscope. So just again to tell you, there's different types of microscopes. The only two types of microscopes I want to talk about is light microscopes and electron, but just know there's even fancier types of microscopes. So this right here is called a light microscope. It's a traditional microscope that we use in lab. It's probably the microscope you've seen all around. Again, it's able to measure things in micrometers. Bacteria are measured in micrometers. If we want to see things like viruses, like if someone has COVID and you take a saliva sample from them or like a nose sample, you cannot use a regular light microscope because viruses are measured in nanometers, and we use electron microscopes to measure viruses. We're going to talk about the transmission electron microscope and the scanning electron microscope in a little bit. Okay, now we're going to focus on the light microscope for a little bit. So a light microscope is any type of microscope that uses regular visible light to look at slides, or we say specimens sometimes. It's just a traditional microscope that we use. And again, it's called a light microscope because it uses regular light. You'll see in lab right here in the microscope, is the light. So here is the light. It goes through this thing called the condenser, and that's why it's called a light microscope. This is specifically called a bright field light microscope. We'll talk about the different types of light microscope, and the total maximum magnification you can get from a light microscope is 1,000 times. So if you want to see something more than 1,000 times bigger, you're going to need an electron microscope. With a light microscope, if I ask you what's the total maximum magnification, it's 1,000 times. It means that you can see something 1,000 times bigger, but that's it. If you need to see something 1,000,000 times bigger, you're going to need an electron microscope. Okay, with the light microscope, we're going to talk about the parts and functions. So let's take a closer look at the light microscope. So the ocular lens, which is also called the eyepiece, is right here, and it has a natural default magnification of 10x, meaning that this, just by looking at it, naturally, you're going to see something 10 times bigger, and it's called the ocular lens. The arm and the base, so this is the arm and the base, they provide structural support so that we can carry a microscope. The objective lenses are these. They're the lenses that magnify what you're looking at, and there's typically four. So for the objective lenses, usually light microscopes have four objective lenses, four magnifications, and one is usually 4x magnification. One of them is 10x, one of them is 40x, and the maximum is 100x. So those are the objective lenses. The stage right here is what holds your slide. Right under the stage, there's something called the condenser. The condenser focuses light on your slide. Here's the light, it goes through the condenser. And on the condenser, there's a little switch or a lever that we call the diaphragm lever. This controls the amount of light entering the condenser. And then the other thing I was gonna say is there's a coarse focus knob and the fine focus knob. The coarse focus knob moves the stage up and down for initial focusing. The fine focus knob makes tiny adjustments to make your image clearer. So these are parts of a microscope. Okay, and how does light pass through a light microscope? After all, it is called a light microscope. So here is your light source. It goes through the condenser, through the objective lenses, and through the ocular piece to your eyes. So that is how light travels. So from the light to the condenser, to the objective lens, to the ocular lens. Again, the ocular lens has a magnification of 10. Objective lenses have different magnifications. What's the magnification of the ocular lens? 10. What is the magnification of the objective lenses? Usually one is 4x, one is 10x, one is 40x, and one is 1,000x. And now I want to talk about something in microscopy that's very important, which is magnification. Before I start off this slide, I want everyone to take note that there is a very big difference between the word magnification and total magnification. So right now I'm going to use the word magnification. The magnification of the ocular lens is 10x. If I ever ask you, what's the magnification of the ocular lens? It's 10x. What's the magnification of the objective lens? It depends on which one you're using. One of them is 4x, one of them is 10x, one of them is 40x, and one of them is 100x. Just to back up a little, I want to make this clear to everyone because sometimes we see these words and we just memorize them. What we mean when we say 4x or 10x, I'll use 4x as an example. It means that it makes something 4 times bigger. If I tell you 10x, it means that makes something 10 times bigger. 40x, 40 times bigger, 100x, 100 times bigger. So the biggest objective lens or the highest magnification is 100x. So that's magnification. So if someone ever asks you, what magnification are you looking at your slide with? They just mean what objective lens are you using? So if you have the 4x objective lens in place and I ask you it's a magnification, 4x. If you have the 40x, you're going to tell me 40x. If I tell you it's the magnification of the ocular lens, 10x. Now we're going to talk about total magnification, which means something completely different. So when I put the word total before magnification, that means the ocular lens, which is always 10, times whatever objective lens you're using. So if I am using the 40x objective lens and I ask you, what's your magnification? It's 40. What's your total magnification? It's 40 times 10, 400x. Now let's talk about what this word oil immersion says here for a little bit. So bacteria, as you know, is really, really tiny. And in order to evaluate it and look at it properly, if you have someone's urine sample or their blood sample and you're looking at it under a microscope, you really want to go to the highest magnification. The highest magnification is 100x. That's a total magnification of 1000x. And at this magnification only, 100x magnification, that's the same thing as saying 1000x total magnification. You have to put oil on your slide, and we call that oil immersion. You don't do it on any of the other slides, and I'll explain it right in a second. So let me talk about resolution before I talk about oil immersion. All I really want you guys to get from this slide about resolution is higher resolution means better image. That's all I want you to get. I don't really mind that you know the exact scientific definition of resolution, but what I want you to know is that when we say something has a better resolution, like we say that this iPhone has a better resolution than the generation before, it means you get a clearer, sharper image. So if we have this image, and this is supposed to be the same thing, this image was taken with a light microscope, and this one was taken with an electron microscope. And I ask you which one has higher resolution, meaning it's clearer, it's the electron microscope. You also get better resolution when you use oil on the 100x objective. So let's talk about that. Okay, oil immersion. So without getting into the physics of it and refractive index, I'm going to try to simplify this as much as I can. Refractive index is just a fancy word for how much light bends. So when you are using the highest objective lens, which is 100x, so let's assume this is your objective lens and this is your slide right here. Let's say your slide has someone's blood sample and you put it under the microscope. The highest objective lens, which is 100, is so close to your slide that when the light goes up, it refracts everywhere. It bends everywhere. That's not good because you won't get a clear image. It'll look very messy. So what we do is whenever we use the 100x objective lens, we have to add oil. You add a drop of oil on your slide. And the reason you add oil is because you get a clearer, sharper image because you prevent light from bending. So oil immersion is only used when we are using the 100x objective lens. That means 100x magnification. That means 1000x total magnification. And again, we use light so that we can avoid refraction or light bending. Okay, contrast. So most microbes are transparent, meaning they're clear. And the best way that I can demonstrate this, and many people watching this have probably had a urinary tract infection or a UTI. When someone has a urinary tract infection, like when you go to the bathroom, you can't just look at your urine and see bacteria swimming around. And there's two reasons. One is we can't see bacteria with our eyes because they're very tiny. And two, it's because even if you have bacteria in your urine, meaning you have an infection, it's clear. The bacteria is the same color as the urine. So we need contrast because most microbes are clear. The way that you get contrast is by using a special microscope or by staining cells, which we will talk about in its own lesson. So right now I'm going to talk about how can we use the microscope to increase contrast, because staining cells sometimes can harm cells. And we'll talk about in the staining section. So there's two ways that you can get contrast with microscopy. These are the types of light microscopes that increase contrast, meaning they're just your regular microscope. There's dark field microscopy. All dark field microscopy means is when you take your regular light microscope and you add a black background. So think if you have a urine sample and you just look at it normally, you're not going to see anything. You're just going to see the urine because the bacteria is transparent. But if you put a dark opaque background, you will be able to see the bacteria. So that's called dark field microscopy, and it's a type of light microscope. It's great because you don't need to do any staining. You don't need to do anything. You're not harming the organism. You're not harming your sample. All you're doing is putting a dark background. That's it. It's like a special disc that you buy that you stick in the microscope that makes the background black, and that's called dark field microscopy. Another type of microscope to increase contrast is called fluorescence microscopy. Fluorescence microscopy uses UV light, and when it uses UV light, it allows you to look at naturally fluorescent organisms. Now, most pathogens are not naturally fluorescent. This is more for people who study like ocean microbes. Or you can also stain microbes using fluorescent staining, and that can definitely harm cells. But if you just care to know what the cells are, you might want to do that. Okay, so dark field microscopy, again, I'll have an image of it. It's exactly what I explained. It's great for looking at live cells without staining them. Urine sample. I love that example. Take someone's urine, you do dark field microscopy. What you do is if you were to just look at it without dark field microscopy, I know this picture looks really clear, but honestly, it's not going to look like that. It's just everything's going to look the same. You just stick a black background and look how great that is. You can see all your organisms in someone's urine. This is not bacteria. This is a huge organism, but I'm just trying to demonstrate that dark field microscopy is great because it lets you see transparent organisms. There's no staining involved and you're not harming the organism. Okay, and then fluorescence microscopy uses UV light. So you use UV light, and if you have naturally fluorescent organisms, then the naturally fluorescent organisms will fluoresce on their own. You can also do fluorescent staining. So a lot of times in medical settings, they do fluorescent staining. It can reveal structures in cells, so it's a powerful tool, and we'll learn more about it later. And just keep in mind, if you do fluorescent staining, like if you fluorescently stain cell organisms that cause syphilis, that's what this image is showing right here, then you are harming the cells. So the impact that these dyes have on cells is you do end up killing the cells. But a lot of the times you don't care if your cells are dead because you took the sample from the patient. You just want to know what it is. But if you're doing it for research purposes, and let's say you want to examine the organisms as they're alive, you want to examine motility, you would not do fluorescent staining. Next is electron microscopes. So everything so far we've been talking about is light microscopes. Now we're going to talk about electron microscopes. So electron microscopes use electrons instead of light to achieve much higher magnifications and resolutions. The reason why we don't see them in lab, well, one reason at least is a lot of times they're huge and they need a lot of work to get set up. So it's not something that you can just have in a traditional lab. There's two main types of electron microscopes, the transmission electron microscope, which we call TEM, and the scanning electron microscope, which we call SEM. Transmission electron microscope is used to look at internal structures and it gives you a 2D image. Scanning electron microscopes gives you 3D images of the surface of a specimen. So electron microscopes are really essential for studying structures that are too small to be seen with light microscopes, like viruses, or even if you want to look at specific structures within cells. So that's what they are. Main thing I want you to get is you can look at things that are nanometers in size, which is like virus size, and you can get a total magnification of up to 1,000,000 times. Remember that with a light microscope, the maximum total magnification you were able to get is 1,000 times. The problem with electron microscope is they require so much sample preparation, so they still have their limitations. Okay, so I don't want you to memorize the extreme details of the difference between scanning electron microscope and transmission electron microscope, but just briefly, transmission electron microscope gives you a 3D image of the scanning electron microscope. Sorry, I want to make sure I said scanning. So SEM, scanning electron microscope, gives you a 3D image, and you can get a total magnification of up to 100,000 times. Transmission electron microscope gives you a 2D image, and the total magnification is you can get up to 1,000,000 times. So that is it for section B of this lesson. Next is section C, which will be on staining.